So hi everyone, and welcome to another Memgraph community call. Today I have Dr. Jason Moore with me, who is chair of the Department of Computational Medicine, Biomedicine, and a lot of else you can see here, and he will definitely present himself. But I must say that I met Jason within the community, Memgraph community, and it was really nice talking to him and his colleagues from Cedars-Sinai, and they were talking to me about their research that is dealing with risk prediction and drug discovery for Alzheimer's disease. And I was quite interested in hearing about that. First of all, like, it's a huge and important topic, I think, and definitely I was interested in how graphs helped in that area. But then when talking further with Jason, I discovered that they are doing a lot of things with also machine learning and LLMs. And today he's going to share with us his research regarding that. And I think that we are going to learn a lot from this knowledge pack session. And yeah, really looking forward to it. And just a small note, feel free to participate. We have a chat, we have questions tab. Feel free to ask questions during this presentation, and I can read them out loud if they are related to something that Jason is currently talking about, and he can respond on the fly. If we skip some kind of question, we can do that after the presentation. Mata will also post a couple of polls, my colleague, so feel free to answer those. And I think that's mostly it from me. I'm looking forward to this because we have started doing quite a lot of community calls on these AI, LLM topics, and I think that a community, Memgraph community, is interested in learning how can they connect their graphs with LLMs, how can I do that, what can I learn from it. So I hope that this presentation will tell us more about that as well. So Jason, the floor is yours. Thank you for joining and thank you for presenting.
Yeah, hello everyone, and thank you Memgraph for the invitation to present to the community. Here's my email address on the slide here, and I'll show it again at the end, but feel free to reach out to me if we can collaborate or work together, or if you have additional questions that we can answer about the work I'm going to present. So I'm an AI methodologist. I develop AI and machine learning methods in the biomedical space, primarily for predicting risk of common diseases. And today I'm going to, the motivation for the work I'm going to present is Alzheimer's disease, which is a neurodegenerative disease and characterized by misfolded proteins leading to lesions in the brain, amyloid plaques, and neurofibrillary tangles, which damage the brain, as you can see in this cartoon, leading to memory loss, impaired thinking, confusion, restlessness, personality, behavior changes, impaired judgment, etc. I think everybody's familiar with the general symptoms of Alzheimer's disease. And unfortunately, this disease is increasing, I think, globally and also here in the United States. And you can see from this graph from the Alzheimer's organization that within 20 or 30 years, the number of people age 65 or older with Alzheimer's disease is expected to double. And this is primarily because the population is aging. We're all living longer, and therefore there's more time to develop Alzheimer's disease. And this is a significant burden for national economies. You can see the figure here in the United States. This year, Alzheimer's disease is expected to cost $360 billion, which is a lot of money. More than half of that coming from our federal and state level organized health insurance, Medicare and Medicaid. So this is a significant population burden and a significant financial burden for different countries to bear. Now, the good news here in the United States is that we have dramatically increased our federal funding through the National Institutes of Health for Alzheimer's disease research. And this fiscal year, we're spending about $3.8 billion on Alzheimer's disease research, which is encouraging. Our Congress has increased federal funding, you can see, pretty dramatically over the last few years, leading to more opportunities for academics and researchers like myself to secure funding to engage in Alzheimer's research. So when I found out that the National Institutes of Health and Congress were putting a lot more money into Alzheimer's, I've worked on Alzheimer's my entire career, but decided to really put more focus on it since there was money available. And this is an $8 million grant that we received from the National Institute of Aging a couple of years ago to develop new AI methods for the study of Alzheimer's disease. And I'm interested in what's called automated machine learning, and we developed one of the very first automated machine learning methods in the world, a method called the tree-based pipeline optimization tool, or TPOT, and I'll show you some results later using TPOT. TPOT's programmed in Python. It's freely available. It's open source. It's on our GitHub. I'll have a link for that at the end. But basically what TPOT does is it pieces together different algorithms for feature selection, feature engineering, feature transformation, machine learning classification or regression, and can piece those together into pipelines to do a particular machine learning task, and can do that automatically and find the right structure, the right operators to do a good job of classification or regression. And so what this grant is focused on is, okay, how do we adapt TPOT to working with multimodal Alzheimer data? We're collecting lots of genomic data, but also imaging data, clinical data, other types of data. And so the challenge is, how do you integrate all this data and develop a machine learning pipeline that can predict Alzheimer's disease risk accurately? And what I'm going to talk about today is using what we call the Alzheimer's disease knowledge base that we developed with the help of Memgraph to inform the machine learning. And one of my concerns about machine learning is that it's often applied to data sets blindly, where the machine learning algorithm is looking for a pattern in the data set, but the machine learning knows nothing about the problem that you're studying. It knows nothing about Alzheimer's disease. It knows nothing about genetics. It knows nothing about brain imaging. And so our goal with the Alzheimer's knowledge base is to inform the machine learning so that it can do a better job of things like feature selection, model interpretation. And I'll just mention my collaborators, Marilyn Ritchie and Lee Shen from the University of Pennsylvania and Philadelphia, are working with me on this project. So about 2 years ago, we developed what we call the Alzheimer's disease knowledge base, or AlzKB. This is publicly available at AlzKB.ai, and we just published a paper a few months ago in the Journal of Medical Internet Research. The citation is there at the bottom of the slide. And I'll show you at the end, AlzKB is open source, and it's on our GitHub page as well. And so the goal of this project was to create a comprehensive knowledge base about Alzheimer's disease. And what we do is integrate more than 20 different knowledge sources about genes, genetic relationships with known genetic relationships with Alzheimer's disease, relationships between genes and drugs and Alzheimer's disease. We also include biochemical pathways, symptoms, and other entities. And so we integrate all those different knowledge sources with biomedical ontology, so we understand the semantic relationships between the different entities. For example, this drug treats Alzheimer's disease, or this gene regulates this other gene. And then we put all of that into a Memgraph Graph Database, and then make that publicly available through the website. So you can go to AlzKB.ai right now, and you can click on the launch button, and that'll take you to the Memgraph page where you can do a Cypher query. So here are the entities that are captured in the biomedical ontology. Most of these I mentioned: genes, drugs, diseases, body parts, symptoms, pathways, and cellular, biological, and molecular function. And you can see some of the semantic relationships here that we capture with the ontology. This is the Memgraph page at AlzKB.ai. And you can see at the top here, we have about 234,000 nodes in the graph and 1.67 million edges or relationships. Now, as all of you know, well know, sorry, I have a typo here with Neo4j. I'll just mention that we started out this project using Neo4j and decided to switch to Memgraph because we believed that Memgraph would give us a performance improvement, which it has. And also, we weren't very happy with Neo4j's very aggressive sales and licensing practices. And so that was those two reasons, the performance boost and the lack of goodwill from Neo4j led us to Memgraph, and so that should say Memgraph in the title there of the slide. So sorry about that, but as you know, the way you query Memgraph or Neo4j is using the Cypher query language, and this is great if you know how to program and can easily learn the syntax. But our target audience is really biologists, clinicians, epidemiologists who don't know how to program, and Cypher would be a major barrier for them. So we want our users, our biology and clinical users, to be able to query the knowledge base and use it without having to learn Cypher. So that led us to thinking about ChatGPT as a tool to help query the knowledge graph. And as expected, we didn't expect ChatGPT to work on this particular problem because it does not understand what is in the knowledge graph that we've assembled, because a knowledge graph really represents a higher level synthesis of all the individual pieces of knowledge. ChatGPT can answer a question about a gene or a drug or Alzheimer's disease, but it doesn't really put all those entities together and understand their relationships in a very complete way. So we found that ChatGPT was no better right out of the box than flipping a coin at answering questions about the knowledge graph. And like I said, that was expected. So about a year ago, we developed a large language model tailored to the knowledge graph, a method we call Kragen. And I'll show on the next slide how this works. And this paper was just published a few months ago, so it's been peer reviewed and published in the Journal of Bioinformatics. And so this is what we had to do to get ChatGPT to accurately answer questions about our knowledge graph. And this is an approach that's pretty standard now in large language models, and I'll explain it in case you're not familiar with it, called in-context learning, or more specifically, what's called retrieval augmented generation, or RAG. And so to get ChatGPT to be able to answer questions, what we do is we take the knowledge graph and we think of all the entity edge connections, we think of those as sentences. And so all the different paths in the graph we think of as a sentence. We convert that to a numeric vector, each of those sentences to a numeric vector, and we put that in a special database called a vector database. And then what you do is you take your ChatGPT prompt and turn that into a numeric vector, and then you use that to query the vector database. And so what you get out is a list of, you basically are doing a similarity matching between the prompt and all the different graph-based vectors that are in the vector database. So what you get out of that is a list of the top hits. You take those top hits and your prompt, and then you send that to ChatGPT. And now ChatGPT has the context. That's the in-context learning part of this. It has the context for the prompt. You're providing it the knowledge that's relevant to the prompt. And now it can generate a natural language response that's more accurate and makes sense to the user. So this is called retrieval augmented generation, and is an example of the more broad in-context learning. So like I said, this is pretty standard in large language models, but this is what you need to do to get ChatGPT to be able to answer questions about things that it wasn't trained on, that isn't publicly available in the text sources that it used. So for example, if you keep a diary beside your bed and make notes about your daily life, ChatGPT is not going to know anything about that. It won't be able to answer specific questions about your daily life, but you could put that in a vector database and then answer questions about it. Now we do one additional big piece to this puzzle to get this to work well. And the challenge of this RAG approach is that questions are often more complex than what can be handled by the system that I just described. So for example, if I have a compound question where I say, show me a list of genes that are related to this particular FDA approved drug and that are associated with Alzheimer's disease, that's a compound question, and the search has to involve two different pieces. You have to search the genes against the drugs and search the genes against the disease. And so you're really looking at different parts of the knowledge graph when you're asking a question like that. So about a year ago, Hugging Face published a paper on arXiv, which I don't think has been peer reviewed and published in a peer reviewed journal yet, on a method they called graph of thoughts. And we read this paper and really thought it would be useful for this task. And so we incorporated it into our large language model approach. And it does exactly what I just described, where you have a complex question or query shown here, list the body parts which overexpress both genes METL5 and STXL2. That's a compound question requiring a compound search. And so what Graph of Thoughts does is it breaks it into two pieces, does the query on the two pieces, puts it back together, and gives you a compound answer. And here's an example of a Graph of Thoughts large language model prompt that we would provide. You are an AI knowledge simplifier with access to a knowledge graph. Your task is to filter out irrelevant information from a given set of knowledge and provide a concise summary. You will receive a statement describing a relationship, and a list of knowledge only includes knowledge directly related to the statement and disregard any extraneous detail. Present a condensed summary knowledge within this particular structure. And then we provided the knowledge from the retrieval augmented generation that I just described on the previous slide. And then you get a response and an answer here. Heart and adrenal gland are the two body parts that overexpress these two genes. So you can see it's fairly complex what we have to do to get the large language model to accurately answer a complex or compound question like this. So not only do we have to do this retrieval augmented generation and put the knowledge graph in a vector database, but we have to do this graph of thoughts and provide these sophisticated prompts with knowledge coming from the RAG to get it to answer the question. So here's the results for this Kragen method, which combines the RAG and the graph of thoughts. So what we did was we constructed about 1,000 true-false questions about the knowledge graph in the Memgraph database, and about 500 multiple choice questions. And you can see GPT-4 right out of the box didn't do much better than flipping a coin at answering these questions, but the graph of thoughts RAG approach did much better. Now we have an enhancement of this method that we call Escargot, which builds on the graph of thoughts, and something called dynamic graph of thoughts, which I'll explain, I think, on the next slide. And this is the title of the paper. It's under review right now. Actually, we just got a request for revisions from the journal, and so we're busy preparing the response and the changes to the paper. The reviewers basically liked it and just wanted to see some more results and had some questions about the method that we will respond to. But here are the results that we showed in the paper. Using GPT here, you can see that it's not performing very well at open-ended questions, true-false questions, and multiple choice questions. The open-ended questions require 1 or 2 hops in the graph to answer, and you can see that it's failing miserably here. Retrieval augmented generation does better, but not very good on these open-ended questions. And then Escargot, which again I'll explain in a minute, is doing much better with about 80 to 95% performance accuracy on answering these questions. So we're getting much better performance. Actually, I don't have a slide describing Escargot. I'm sorry about that. But basically what Escargot does is make the graph of thoughts more dynamic. And I think this will be of interest to Memgraph. Is what we actually do is take the query, we break it into pieces, and then from those pieces, we write Python code to perform a cipher query of Memgraph directly instead of using the vector database with the RAG. We can actually break this down into pieces, and on the fly, this is the dynamic part of this, we can write Python code, do the cipher queries, and then bring the pieces back together to answer the question. So it's more dynamic in the sense that we're generating code on the fly and creating this graph structure on the fly to look at all the different pieces and parts of the complex question and then piece it back together. So that's what the dynamic graph of thoughts does. Okay, so now maybe I'll stop there, and Katerina, are there any questions at this point?
So currently we don't have any, but maybe I have one. So I would maybe like, can you explain? You now said that now you don't, within Escargot you wouldn't have a vector database, but previously you had a vector and a graph database. What made you have a graph database in the first place? Like, what more do you get with data stored within graph and then embedding it and storing it into vector, as opposed to like some basic RAG where people just utilize vector databases? What do you get with this graph structure?
Well, the Memgraph graph database is extremely important for this particular problem because the ontology and the graph really describe the higher order relationships between all the entities that we're interested in. And that knowledge is extremely important, and that's why, you know, ChatGPT can't answer questions about it, because that knowledge doesn't exist in the public domain in a searchable fashion and in a way that the ChatGPT large language model could have been trained on, because it's a higher level synthesis. Each of the knowledge sources individually is an important source of knowledge that, again, ChatGPT doesn't really have access to. And we're piecing together 20 different knowledge sources and using the ontology to model the semantic relationships between the entities, and then putting that into the graph form. So the answer to your question is that the graph gives us a higher level synthesis of the knowledge than we would get in any other way. So the graph is a very, very important part of this. Mm-hmm. And then the tools that I've described ultimately allow us to very quickly, efficiently, accurately query that knowledge from a ChatGPT prompt. Does that make sense?
Yeah, it does. So we actually had a similar question now from Jeff, saying like, does the tool allow for natural language questions that translate automatically into knowledge graph queries?
That's right. So the user enters a natural language query, and our AlzKB website, I don't think it's live today, but in the next few days or by early next week, you will be able to, with a paid ChatGPT account, you'll be able to log in and enter natural language queries through a ChatGPT interface. And those queries, using our Kragen and Escargot methods that I just described, will then do the query and provide the answer back in natural language. So yes, very soon you'll be able to log in through ChatGPT, through our AlzKB website, through the ChatGPT API, and ask natural language queries of the knowledge graph in Memgraph and get natural language answers back. So we're pretty excited about this, and we think this is going to make the Memgraph graph database much more user-friendly for our users.
Yeah, that sounds really cool, and sounds, I think, like a dream to anyone, especially those who don't know Cypher. We also have a question, and we have more questions, so hope you're ready. How much, and if at all, does performance really make a difference to your use case? Like, are there any aspects of real time that play a role for you? How much is the performance important?
I think performance is pretty good. Within seconds, we are getting answers back. So performance so far hasn't been an issue. I think it helps having the Memgraph graph in memory. That certainly speeds up the queries, the Cipher queries on the back, that Escargot is doing on the fly. And our knowledge graph is not outrageously large. It's probably small on the smaller side compared to some of the knowledge graphs that some of you might have developed.
And does that graph grow with time, or is it like a static one?
It will grow with time as we add more entities. We don't have a huge number of new entities to add to the graph, but we do plan to add some more, and that will add a lot of new edges and increase the size. But I still, even with those few additional entities that we have planned, I don't think it's going to grow. It'll be well below the memory limits of most people's computers or laptops that are using this.
And we have a question. So Bassem said that you have outlined an excellent process with what you said, and he asked, can this be generalized to other type of diseases, so not only to Alzheimer's?
Absolutely. We have an addiction knowledge graph that we're about to release. We're building one for ALS, which is another neurodegenerative disease, and we have several others planned. So it's open source. It would be easy to go and you could swap out in the knowledge graph Alzheimer's for whatever disease you're interested in, and it really is almost that easy. So yes, this can be specialized for any particular disease or can be generalized for multiple diseases.
That's really nice, yeah. And how critical is having a right ontology for the RAG accuracy? Do you know?
Oh, that's a great question. We haven't looked at that specifically, but I do think I'm not sure how important it is for the accuracy of the questions that are based on the knowledge graph, because the questions shown in the table here on this slide are all derived from the graph. And I think since they're derived from the graph, however you've constructed the graph will define the questions, and then the ability of Escargot or RAG to answer the questions just depends on what methodology you're using. But I think the more general question, if I can build on it, is to say how accurate are the answers for Alzheimer's disease, and how relevant is the knowledge that you're capturing with your query, how relevant is it to the study of Alzheimer's disease and informing, say, experimental studies or clinical studies? I think that's the more important question. And for that question, I think the ontology is very important, because that really defines the relationships between the entities, and that the accuracy with which the ontology defines those entity relationships will make the results either more or less relevant to Alzheimer's disease specifically. I hope that makes sense.
Yeah, I think it definitely depends on the type of questions you tend to ask and the use case you're planning to work on. We have another question. So how rich is the schema of the graph? Are you making use of multiple properties on both nodes and edges?
Yeah, that's a really, really great question. Thanks for asking that. We have right now our graph is relatively simple, but we do plan. We have started a project to enrich the properties of the nodes and the edges with more information, maybe inferring probabilities of relationships and those kinds of things. So we definitely plan to so-called color the graph with more information, and that's in the works. And I think that will add a lot of value to the graph. So thanks for asking that.
Mm-hmm. And just two more questions,
Sure.
and then maybe we can continue. So first, how do you quantify and solve the uncertainty in knowledge graphs and inconsistency among different databases? And yeah, I will want to read on that.
Yeah, well, that's a great question, and there is uncertainty in each knowledge source, and obviously the quality of our graph is only as good as the knowledge sources that we're pulling from and the ontology that we've constructed. So there is uncertainty, and I guess I'm not an expert in quantifying that uncertainty, but I agree that's something we should probably be doing. I think it'd be wonderful to be able to express to our users the degree of uncertainty in any edge relationship or in the graph overall. I think that would be really important, but like I said, I'm not quite sure how to do that in a meaningful way, because I didn't develop the underlying knowledge sources that we're using. We have to trust that that they're good knowledge sources, that they're reliable. And what I would say is that we are using knowledge sources, most of which have been around for years, have been peer reviewed, published. Many research groups have used these knowledge sources. There are lots of publications with the knowledge sources. That doesn't mean there is an uncertainty, but I think, to some degree, we're trusting that these are well-run, well-developed, constantly updated knowledge sources that we can trust to some degree.
Yeah, and another, like the last question until you continue. So how to dynamically update knowledge graph database and ensure its completeness?
So we update our knowledge graph for AlzKB monthly, or at least every few months. As the underlying knowledge sources change, we refresh the graph and create a version number, so that it is constant. We do have a team of people constantly updating this, at least on a monthly or bi-monthly basis. But not
I would say also, to Elvis, who has the question, like, it's definitely not an easy task if you want to do it that more often. But with Memgraph, you can dynamically update the graph with triggers, and you can have data coming in with different streams. And there are also online graph algorithms, like dynamic ones, that help you recalculate the values that you might be using in a graph. But it's definitely an interesting process, having also like a dynamic knowledge growing all the time in real time. So I think we'll be seeing a lot of those kind of use cases in the future as well.
Yeah, thanks, Katerina. Okay, I'll move on, and I think there'll be time for questions at the end or a little bit later. So now I want to show you as an example of how we're using the knowledge graph and the large language model tools that I've talked about for the genetic analysis of Alzheimer's disease. And there have been a lot of large-scale genetic studies of Alzheimer's, and there are about 100 genes. There are about 20,000 genes in the human genome, and about 100 of those have been identified as risk factors for Alzheimer's disease. An example is the apolipoprotein E gene, which is a lipid metabolism gene, which is a very strong risk factor for Alzheimer's. So there are about 100. And what my goal is to identify new genetic risk factors that have been not found before, and ultimately new drugs for Alzheimer's disease. I think I forgot to say earlier, one of the rationales for our Congress in the United States for putting hundreds and hundreds of millions of dollars of new money into Alzheimer's research is to ultimately lead to new treatments, new drugs for Alzheimer's. So I firmly believe that those 100 genes, lots of people are working on those 100 genes and their protein products and developing new drug targets. And you could think of that as kind of looking under the lamppost, where each lamppost here is a different gene that's been found. What I'm interested in is looking across the street and identifying new genes and new drug targets and new drugs for Alzheimer's disease. I'm more excited about the novel discoveries than I am about looking under the lamppost at the known 100 genes, because lots of people are working on those. And so I talked earlier about automated machine learning, that we have this method for automatically building machine learning pipelines. And what I'm interested in is how can we use our graph database and the knowledge graph that I've talked about to inform feature selection for the automated machine learning pipelines that are being developed. And you could think of each of these clusters here. The gray may be an FDA-approved drug for any disease, and the blue are the genes connected to those drugs. And that's kind of how we're thinking about the knowledge. Using the knowledge graph is to define, look for drugs, look for genes, use those as feature sets that can be then selected automatically by our automated machine learning algorithm. So let me just step you through some cutting edge results that are not published yet, but so please consider this a work in progress. So we have access to a very large data set for about 30,000 human subjects from what's called the Alzheimer's Disease Sequencing Project, which is a NIH-funded large-scale project of collecting data on Alzheimer's disease here in the United States. And so we have genetic measurements across the human genome for these 30,000 subjects. We split that into a training and testing set. And then we create what's called a gene risk score for every one of the 20,000 genes from all the genetic measurements. So that's the data that we're working with in this study. And then we did two additional things. We focused on protein coding genes, so that limits the gene set to about 20,000. And then we further limit it to just those genes whose protein products are believed based on their chemical structure to be druggable, which means they have a chemical property that where a small molecule could be developed to target part of the protein to influence it. And there are about a couple thousand genes that are considered druggable based on their chemical structure. So the data set is about 30,000 rows of human subjects with and without Alzheimer's disease, and then a couple thousand columns of features or variables that we're using. And we want to develop a machine learning classifier based on the couple thousand features, the couple thousand genes for predicting who has Alzheimer's disease versus who doesn't. So that's the goal here. And so what we did was we wanted to, and again, I don't want to look under the lamppost. I want to look across the street in the dark spaces for novel discoveries of genes that could lead us to new drugs. So what we did, and this is I think an interesting way to use the knowledge graph, is we created what's called a variational graph autoencoder. This is a deep learning algorithm that takes the graph structure as input and then embeds the graph properties in hidden layer nodes. And then because it's an autoencoder, then using those hidden layer nodes, it tries to recreate the graph. And the hidden layer nodes we use as embeddings in multi-dimensional space. So this is an example in 2-dimensional space, but where the results I'm going to show are in 16-dimensional space. The red are the known 100 Alzheimer genes, and then the other colored are the rest of the genes in the data set. And so again, I'm not interested in this area here because the red area, because those are the known Alzheimer genes and presumably things in the graph that are closely connected to the Alzheimer genes. I think the novel discoveries are going to come from this part of the embedding space that's further away from Alzheimer's disease. And then the other thing we can do is we can create drug embeddings. So here are the known FDA-approved drugs for Alzheimer's disease. And again, I'm interested in the drugs that are not related to Alzheimer's disease, because those are drugs that potentially could be repurposed. You know, these are drugs that are used to treat other diseases, and they could potentially be repurposed to treat Alzheimer's disease. So I'll show you in the next slide how we're using this embedding information, but this is all based on the graph. And so we're using the graph to point us to novel regions of gene space and drug space where we can focus our machine learning algorithm. So this is a new method we've developed using a published algorithm called Map Elites, and we've extended it to do this novel feature selection using the graph. So here we have our Memgraph Graph Database, and we're using this in two different ways. We're using this as as I said in the last slide, to define distance away from known Alzheimer genes here, and I'll explain how this works in a minute. And then over here, we're using the drug information to define drugs that are not related to Alzheimer's disease and the genes that are connected to them. And by creating feature sets, variable sets that are tied to FDA-approved drugs, when we find something interesting using the machine learning, then we can go back. We know it's a gene set tied to an FDA-approved drug, and then that drug becomes a candidate for being repurposed for Alzheimer's disease. So that's why we're organizing it in this way, to make the machine learning results more interpretable. So let me just say this again. The knowledge graph, the Memgraph database, is being used to guide the machine learning over here toward novel parts of the search space, novel genes, and it's being used here to define novel feature sets tied to FDA-approved drugs, so that that'll make the machine learning more interpretable. So up here we have our automated machine learning method that generates thousands of different machine learning pipelines with these different feature sets. I think there are about 500 or 600 feature sets that it can choose from, tied to the FDA-approved drugs. It generates lots of pipelines. And then what we do is we map the thousands of pipelines to this two-dimensional space here, where the numbers here represent the distance from known Alzheimer genes. So things down here in this part of the graph, in this part of this plot, are things that are close to Alzheimer's disease in that embedding space that I showed. And then what I'm interested in are the things up here, which are things that are further away from known Alzheimer's disease genes. This is where the novel discoveries are going to come from. So this map elites method maps all the different machine learning pipelines into this two-dimensional space, things that are close to Alzheimer's, things that are further away from Alzheimer's. We pick the elites, the best in each of these squares. We pick the best pipelines, and those pipelines are used to generate new pipelines and generate variability. And that's how this, and then the machine learning iterates through lots of different machine learning pipelines using this feature space. So let me show you what we get at the end. Here's an example result after running this algorithm. The number here represents the feature set. And I said there are up close to over 500 different feature sets, so that's what the numbers represent. The brown and yellow are the best performing machine learning pipelines, and so you can see the color scale here. So this is the area under the ROC curve, so bigger is better. And what was interesting, and this is exactly what I wanted to discover, is that we got a feature set here at the top, number 359, that I'll explain in a minute, that had an area under the ROC curve of about 0.59, and it performed just as well as the things feature set here, 476, which I'm not going to show you, which are things that are closer related to Alzheimer's disease. So this was encouraging to us. The algorithm was able to, using the knowledge graph, automatically discover a feature set, a gene set that performed just as well as things we know about for Alzheimer's disease. So here are the genes, and I know this won't mean much to most of you. And the drug here was temazepam, which is a drug class from a drug class called benzodiazepines. Temazepam is actually prescribed for insomnia. It targets neurotransmitter genes. These GABR are GABA receptors. This is a class of genes that code for proteins that are involved in neurotransmission. So the more active these genes are, the more heightened state your brain has. And what temazepam does is it dampens the signals through those GABA receptors in the brain. It dampens your neurotransmission and calms your brain, and that's why it's prescribed for insomnia and helps you go to sleep. And actually, temazepam is prescribed to insomnia as a symptom of Alzheimer's disease. A lot of Alzheimer patients can't sleep very well, and so temazepam and other neurotransmitter drugs are prescribed to sort of quiet the brain and allow you to sleep. Ibuprofen was a second drug that also was in this group from the knowledge graph, which is an anti-inflammatory, and inflammation is a big part of Alzheimer's disease. So this is interesting because these genes are not directly tied to Alzheimer's disease. In fact, they're quite far away in the knowledge graph from Alzheimer's disease, but they are brain related because they affect neurotransmission. And so this is just some gene interpretation that we did. this is using a resource called HumanBase, which looks at the experimental evidence for relationships between genes. This is a derived graph. This is not from our knowledge graph, but this is from a derived graph from this HumanBase, looking at the relationships between the genes. Here's the GABA receptor genes. Cytochrome P450 is a drug metabolism gene. And these are the pathways that were found: drug metabolism, which is from here, and then these chloride channel receptor metabolism is related to these GABA receptor genes because they're found in the cell membrane and are involved in chloride transport. Now, what was really interesting to me is that if you search on temazepam and Alzheimer's, you in the PubMed database of published biomedical research articles and clinical articles, you only find 2 papers. And so this is not a drug that is used to treat Alzheimer's disease, which is what I'm after. It's a drug that's used to treat Alzheimer's symptoms or insomnia. So so this makes it a more interesting result to me. So I'll stop there. And so we've done, I think, 2 important pieces of work here. We've shown how we can tailor large language models using some novel tools that we've developed to accurately query the Memgraph database, the knowledge graph, and we've demonstrated how we can use the knowledge in the knowledge graph to inform machine learning to get at novel results for Alzheimer's disease. Now, what we're working on right now, and we have a prototype working and hope to roll this out in the coming month, 2 or 3 months, is a completely automated system for doing machine learning informed by the Memgraph knowledge graph, where you can just talk your way through an analysis. You can say, show me the genes tied to this drug and this disease. It'll give you the list. You can say, create a data set with only those genes, those features. The data set gets created. Then you can say, perform machine learning on this data set. The automated machine learning takes over and does the machine learning. And then you can ask for an interpretation. You could ask for, say, Shapley values or feature importance scores based on the machine learning result, and it will show you the most important genes, the most important features. So large language models and the automated machine learning work that we've done and the Memgraph Graph Database that we've developed allow us to now completely automate through natural language all the steps of doing a machine learning analysis. So I'll stop there, and I think there's a few more minutes for questions. This is the Teapot team, my software engineers that have done an amazing job on the automated machine learning. Joe Romano was a postdoc in the lab that created the ontology and the graph database that led to AlzKB. He's now an assistant professor at the University of Pennsylvania. My software engineering team did all the work on the Kragen and Escargot large language models. Nick Matsumoto in particular is really the brains behind the operation and came up with these amazing strategies. Sissy Xiao's a graduate student in my lab that is doing all the map elites, using the graph database that I talked about. Here's my Alzheimer grant that supports all the Alzheimer work. Our GitHub pages with all the open source software, including Kragen and Escargot. If you're interested in adding a natural language query component to your Memgraph database, you could adapt these to, I think, pretty easily to what you're doing. And AlzKB is also open source. Someone asked a question about extending this to other diseases, which you could adopt this, change the ontology, the knowledge source to include whatever disease you're interested in, and go from there. And you can use this however you want. Thank you to Memgraph, and I'll stop there and take more questions.
Thank you, Jason. Yeah, this was really interesting, especially the last part. I mean, hearing about your conclusions regarding those medicines, I think it's quite a big thing, and it's quite a good way of thinking to trying to discover something that's not under the spotlight. And having graphs help there is just like it's just amazing to hear that and to hear about your approach. So yeah, kudos on the great work for your team and yourself, and great work, everyone, definitely.
Thank you.
I shared the links of all of the open source projects that you mentioned within the chat because the participants also asked for it. So I think they're really interested in learning about the project. I see some of them are typing. Yeah, you have thank you, that it was great. It was really clear explanation and presentation, and another kudos saying that it was very impressive work, and thank you from everyone. I think that everyone is quite satisfied with the presentation. My question maybe would be regarding these medicines that you explored and these genes. Are these medicine being explored as like a preventive way of doing, like to not have Alzheimer's disease, or to kind of cure it if you already have it? Like, what's the end goal? Is the goal to kind of not even get it in the first place?
Well, this, because we're specifically modeling risk of Alzheimer's disease and using people with known Alzheimer's disease to train our machine learning model, I think any drug that we find using this method would be a candidate for treating somebody already with Alzheimer's disease. So I think to get at prevention, I think we would need to train on a data set of maybe early Alzheimer's disease. And late Alzheimer's disease, and specifically model the progression of Alzheimer's. And I think drugs found in that way could be more preventive in nature. But it's possible some of the drugs we discover could be preventive. But I mean, there's a tremendous amount of interest, of course. I think your question's a good one in, detecting early Alzheimer's disease and preventing Alzheimer's disease in the first place, or preventing progression once you've been identified as having some memory issues or some early symptoms of Alzheimer's, and just stopping it. Some of the drugs that there just aren't good drugs for Alzheimer's. Some of the drugs, there have been several FDA approved drugs that have been approved in the last few years, and they're not very good. They do slow Alzheimer's disease somewhat, but we're still looking for that blockbuster drug that's really going to have a large impact on this disease.
Yeah, and I'm looking forward to following up with your research, and I believe it's going into a good direction. And yeah, definitely great work, everyone, and especially with focusing in the future on other diseases as well. I think this could be really huge. Also, a message from Jeff saying, like, best of luck with your research, and thank you for sharing your work, and well done on the presentation. So I definitely agree with Jeff. So yeah, I think that we can wrap up. I think that your presentation was really nice, Jason. Thank you for giving us a great introduction to this topic, because myself, I knew what you were doing high level, but you gave a really good overview for me to understand also some specifics that I can about the topic, and I think you did that pretty well. And thank you for sharing this knowledge with the community as well. Yeah, do you have anything else to say?
Oh, just thank you to Memgraph. We really enjoy your product and have found it very useful. And thanks to the community for all the wonderful questions. I appreciate it.
Yes, thank you, Jason, for participating here, and thank you everyone who joined, and see you all very soon on another Memgraph community call.
Bye-bye.
Bye, everyone.